Okay, the isotonic, hypotonic, and also the hypertonic solutions. So, what is the meaning of iso, hypo, and hyper? Iso, the same, hypo, less, and hyper, more. Okay, so actually, what is the meaning of tonic or tonicity in biology? Okay, so this is what I found from the internet. So it says tonicity is a measure of effective osmotic pressure gradient. Okay, so that means the water potential between the two solutions which are separated by the semi-permeable plasma membrane. So in other words, it is referring to the relative concentration of solutes in a dissolved solution, and that's going to determine the direction or the extent of diffusion in which we will be looking at it. So still remember the terms osmosis and the passive transport. Okay, it is referring to the movement of the water particles. So let's look at the first concept over here, the isotonic solution. So we have here in the middle the selectively permeable membrane, which is going to separate this solution into two compartments. So we have two compartments for us to compare the concentration of the solution, and we name it as solution A and solution B. So since this is isotonic solution, solution A and B have the same concentrations of solution. So that means we can say that solution A and B are isotonic towards one another. Okay, so first thing to do, the first step is that you compare the concentration. Okay, now that you have known that they are isotonic towards one another, second question is that you ask, is there any net movement of water? So in this case, since having the same concentrations of solutions, therefore there is no net movement of water. Now you look at the arrows over here to the right and to the left. So that means the water particles, they are diffusing across this selectively permeable membrane at a constant rate. Okay, so that means even though at isotonic solution, it doesn't mean that the water particles is static. They are not static, they are still moving. So that's why we say they have reached dynamic equilibrium. Dynamic means moving, still moving. Okay, so you can also double check and count. Look at these water molecules, okay, depicted by the blue oxygen, and these two over here, they are the hydrogens, H2O. So the big ones, the red ones, they are the solutes. So if you are to count this one, A, solution A, the number of water molecules is 8. So double check at solution B also. Yeah, the number of water particles is 8. So that means they are isotonic towards one another. So how about hypotonic solution? Hypo. Okay, hypo means less. So in this case, it is referring to less solutes concentration. So for solution A, when you look at it, you can see straight away solution A has a low solutes concentration and high water potential as compared to solution B. So in other words, we can say solution A is hypotonic to solution B. Okay, having identified the concentration, the next step is to ask, is there any net movement of water? So in this case, yes, there is net movement of water. Then the next question is from where to where? So osmosis tells us that water will diffuse from region of high water potential to region of low water potential. That means it is down the concentration gradient without the expenditure of energy. So without the use of energy, it's a passive transport. Therefore, water diffuses from solution A to solution B by osmosis. Don't forget to include this term when you answer your structure or your essays questions. There's marks given for this. Okay, so the third solution is hypertonic solution. Hypertonic, so that means high solutes concentration. Okay, you can see right here in the picture, and having low water potential. And osmosis tells us that water will diffuse from region of high water potential, in this case that is B, to a region of lower water potential, and in this case is solution A. So the first step is to identify the concentrations of the solution by saying solution A is hypertonic to B. And the next question is to ask, is there net movement of water? Yes. So you say from where to where, and you add the word by osmosis. So in this case, so here's what we are going to do. Water diffuses from solution B to A by osmosis. Okay. Okay, the effects of hypertonic, hypertonic, and isotonic solutions on animal cells and the plant cells. Okay, so this one, some biological lens over here, the term hemolysis. Okay, this is the one, hemolysis is only used for the burst of the red blood cells, because the term heme over here comes from the word hemoglobin, and that's the pigment that gives the red blood cells the color red. So that's why it is exclusive and only to be used for the burst of the red blood cells. Okay, so here let's look at the sentences, the statements. It says the cell cytoplasm contains solutes such as glucose, mineral salts. So the fluid, okay, take note, which is in the cell, we call it as the okay, inside cellular cell, so intracellular fluid. And each cell, it is surrounded by the extracellular fluid. Okay, take note of the terms. Therefore, cell will always go through osmosis process, because inside and outside the compartment has got water. Okay, so they will always go through the osmosis process and the movement of water across the plasma membrane. So whether it depends on the water potential of these two regions. Okay, so that's bring us to this slide, which we will be discussing on the red blood cells. Okay, the effect of hypotonic solution. So imagine these red blood cells placed in a hypotonic solution. Okay, so that means now we are comparing the intracellular fluid and also the extracellular fluid. Now, in this case, this red blood cells is now surrounded by hypotonic solution. Okay, hypotonic solution. So red blood cells is hypertonic to the solution here, the extracellular solution, which is hypotonic. So the next question is, you ask yourself, yes, in this case, there is net movement of water. Now you ask yourself, from where to where? So very simple, you just ask yourself, which compartment has a higher potential of water, higher water potential? Okay, since red blood cells is hypertonic to the surrounding solution over here, meaning this solution, the surrounding solution, is now having higher water potential. Therefore, there will be net movement of water. Water will diffuse into the cell. Into the cell, this compartment has got higher water potential. So into the cell, don't forget to put the word by osmosis. Eventually, this cell is going to swell and finally burst. Burst. Why burst? Because the plasma membrane is too thin to withstand the osmotic pressure built up in the cells. Unlike the plant cells, which has got the turgid cell wall, the tough cell wall. Okay, for red blood cells, the plasma membrane is thin. There's no cell wall. Okay, so the burst of the red blood cells is known as hemolysis. Lysis comes from the word lys, which means break, pecah. Okay, hemolysis. Okay, next is that imagine now the red blood cells is placed in a hypertonic solution. Hypertonic. So outside, inside, the first thing to do is to identify the concentration. So if it is in a hypertonic solution, okay, meaning here has got low water potential as compared to the intracellular fluid. So there will be net movement of water where water will diffuse, use the word diffused instead of move, diffuse out of the cell by osmosis. Do not forget osmosis, which will cause, now this is the observation, the cell to shrink. And there's another term that you need to know for the red blood cells over here. It is now undergoing crenation. Okay, crenation. So the third condition, if the red blood cells is now in a solution which is isotonic to it. So that means the water potential inside and outside the cell is now the same. So definitely, very obvious, there's no net movement of water, which means water will diffuse into the cell, out of the cell by osmosis at the same rate, which means they have achieved dynamic equilibrium. So you see the arrow here, in and out, the water molecules at the same rate. No net movement of water across the plasma membrane. Therefore, observation, the cells maintain their normal shape.